I am presenting on June 20th, 2026. Auto-islet transplants treat patients with chronic pancreatitis. A surgical team removes the diseased pancreas and transports it to a clean room facility where a team performs benchtop surgical work to clear away the junk, dissect it to cannulate and inject the ducts with an enzyme mixture down while releasing the islets, collect as many islets as possible, and prepare IV bags to infuse them into the patient's liver portal vein for engraftment, where the patient can still produce insulin independently. For islet allo-transplants to treat type 1 diabetics, the steps are the same, except that the pancreas is from a healthy deceased donor. So type 1 diabetic pancreases are not good for auto transplants due to the abnormal extracellular matrix, where it's harder to break down the pancreas and it's trapping the islets, a smaller organ size, likely from the hypoxic environment created by autoimmunity, and the islets lacking beta cells that produce insulin. The FDA classified islets for allotransplants as a biologic drug, but islets for autotransplants are not biologic drugs. The FDA requires the transplant centers to sponsor a biological license application, or BLA, before clinical use, stalling 20 years of development in the field. A private for-profit group, Celltrans, submitted theirs and recently got approved, allowing commercialization of islets. It's inconsistent with the prohibition of selling human organs according to the National Organ Transplant Act. This law also created the Organ Procurement and Transplantation Network, operated by United Network for Organ Sharing, to oversee organ matching. Without oversight on the islets, risking safety and effectiveness of the allotransplants, the islets are less regulated. Celltrans will have 7 years of marketing exclusivity under the Orphan Drug Act, a U.S. law from 1983 that incentivizes pharmaceutical companies to research and develop treatments for rare diseases. Significant leverage in terms of contract with any transplant center to provide islets at a cost, and dictation over which transplant centers offer the treatment. Transplant centers will have no alternative source of islets, and there will be no quality control. At least 50 islet experts classify islets as microorgans because islets are not artificially manufactured. Islets are a cluster of cells with their own function, and they have their own circulatory network. They maintain their own morphology during the processing and post-transplantation. They connect their own vasculature to the recipient's liver portal vein for engraftment. Islets can't handle below zero temperatures and are only preserved at room temperature for a short time, making it impossible to have islet cell banks. And the outcomes depend on numerous parameters instead of a single test. The sterility of the environment and team members, and constant supervisions of donor selection, processing, infusion, and post-transplant care. The only way to know the potency of the islets is not through a certificate, but with how well a patient responds. Academic centers performed allo transplants without the BLA for the past 20 years. Afterwards, islet centers islet allotransplantation is not broadly available to American type 1 diabetics, while in other countries, islets are seen as organs or tissue, and the treatment is a standard of care. Ironically, these early programs benefit from the American-led research. The issue of influential view is the inconsistency with the National Organ Transplant Act and patient outcomes regarding lack of quality control of the islets and post-transplant monitoring from OPTN or UNOS. It also limits the patient's access to treatment and risks jobs of transplant team members. From a utilitarian approach, more American type 1 diabetics will be treated without transplant centers sponsoring a BLA and without the increasing costs. From a deontological approach, classifying islets as biologic drugs limits patient access and limits their outcomes. The autonomy is limited due to companies dictating which transplant centers offer the treatment for the cost they come up with, and what providers can treat their patients with, further limiting their access. This dilemma fails to follow beneficence, non-maleficence, and justice principles by preventing beneficial therapy. Islets should be free of contamination when companies follow current good manufacturing practices, but patients are still exposed to uncontrolled clinical risks. With the fidelity principle, islets, naturally varying, cannot be standardized as drugs. Celtrans submitted a check their safety, purity, and potency of the islets. They'll need to check the viability and function through glucose stimulation insulin secretion assays before releasing islets to transplant centers. The other reason is for risk assessment, since patients undergo immunosuppression before and after allo-transplants to prevent graft versus host disease. The FDA argued that islets for allotransplants are not analogous to whole pancreas transplants, while cell demise, safety of the islets changes over time and are fragile at certain temperatures. There may be delays from glucose stimulation, insulin secretion assays, glucose stimulation index. Cell transplant be responsible for screening the donors, like their age or BMI, underlying conditions, and infections, possibly leading to donor scarcity. While islets for allotransplants are not analogous, they're microorgans, and while the FDA is concerned about tissue manipulation, the steps are the same for auto transplants. The only difference is where the pancreas came from. In the end, islets are islets. I and many islet experts suggest that the FDA should reconsider their illogical classification of islets for allotransplants as biologic drugs and the requirement for transplant centers to sponsor a BLA, and instead think about the many people who desperately need this treatment.